Welcome to the third module for the Global DARE Foundation Patient Collaborator Training Series. My name is Mousumi Bose, and I will be your narrator throughout these training modules to help you become an active patient collaborator in ongoing and upcoming research for adult refsum disease. In this module, we are going to be focusing on this important concept of the natural history of a rare disease. We're going to talk about what natural history is, what information natural history gives us, and how it is useful to the rare disease community, and what role patient collaborators within a rare disease community play in better understanding the natural history of their disease. We will then talk about what are the different ways to study natural history, and we will close with some case studies of rare disease communities that have gathered natural history data and what impact it's had on their patients. Let's take a second and define what natural history means. Obviously, in this case, we're not talking about the history of nature. When we are talking about the natural history of a disease, the official definition is the course that a disease takes in the absence of intervention in diagnosed individuals. And the timeline for this course of natural history goes from the onset of the disease or just before until the disease is resolved or until the death of the individual that has the disease. Simply put, the natural history of a disease refers to the progression of a disease process in an individual over the course of their life over time. So what this graphic is meant to show is that the natural history timeline spanning from before the exposure to the disease, and I will use this moment to say that because the majority of rare diseases are genetic by nature, that exposure occurs in utero when an individual inherits or has acquired in another way a genetic variant that is disease causing. So natural history starts there or just before there. Once that exposure occurs, natural history is an understanding of everything that happens related to the disease through the subclinical or pre-symptomatic stage of the disease, where an individual may not experience symptoms at that time, but there may be environmental factors like diet or just even time that contribute to the onset of symptoms. Through the actual onset of those symptoms, through the time that the disease has fully manifested from a clinical perspective, through the time that a person has usually succumbed to the disease. Unfortunately, with genetic disorders, the disease isn't actually resolved because the person has that genetic disorder throughout the course of their life. So the natural history of that disease in that person ends at the end of their life. So I mentioned that natural history is an understanding of everything that occurs during that timeline that I presented in the previous slide. So let's get a little bit more specific about what that understanding encompasses. Natural history can give us information on how a disease develops over that timeline from exposure through symptom onset through the course of an individual's life, how it manifests in that person, and how various factors, whether they be genetic or environmental or otherwise, influences how that disease manifests. And information on natural history can be collected in a variety of ways. One common way is through the medical evaluation of a patient. Another way to collect natural history information is through the patient reporting on their own lived experiences. Finally, the patient can also provide samples of tissue or blood to be evaluated for natural history information. We'll get into the specifics of natural history data collection, particularly looking at this clinical evaluation and patient reporting shortly. So I wanted to start with showing an example of a disease that has a well-documented natural history. This illustration is representing the natural history of type 2 diabetes. So type 2 diabetes is a condition that is caused by both genetic and environmental factors. Prior to diagnosis, the patient becomes increasingly more resistant to the effects of insulin, which results in high blood glucose or blood sugar levels. This requires the pancreas to work harder to pump out more insulin. Once a patient gets diagnosed, the degree of insulin resistance remains relatively unchanged, but the pancreas keeps working overtime to ensure that there is enough insulin to keep blood glucose levels down. Eventually, the pancreas gives out due to exhaustion and stops producing insulin altogether, and this causes blood glucose to go even higher than before, eventually causing damage to peripheral organs. So here is a disease where we now know what happens to a diagnosed patient over the course of their life. And it's this kind of information that was instrumental in the development of drugs like Ozempic, which, as you probably know, has been a game changer for people living with diabetes and obesity as well. So this example is meant to demonstrate why natural history is so important. When you have a comprehensive understanding of a disease and its clinical course, it allows you to potentially uncover new aspects of a disease that were previously not well documented. And this may allow for better diagnosis as well as identify potential new therapeutic targets. And in some cases, like when an already approved drug is being used for a different disease or a different outcome, and this happens frequently in rare disease, natural history can be used to identify symptoms or biomarkers that might be useful in testing the effectiveness of these repurposed drugs. So collectively, this can give rise to more clinical trials, which can lead to more commercially available treatments that could ultimately improve the prognosis and the quality of life for patients that have this disease. So these are some of the ways that natural history can be used. And if we're going back to this question of why is natural history so important, specifically in the context of rare disease, here's the thing. Most rare diseases have little to no documented natural history. So let's expand on that some more. So many of us rare disease advocates are very familiar with the talking point that 95% of all rare diseases have no FDA-approved treatments. And the reason that there are so few treatments is because the number of current clinical trials for rare disease are few and far between, relatively speaking. One big reason for so few clinical trials is probably related to the fact that the technology for new treatments is just not there yet. This is definitely the case for things like gene-based therapies, but another really big reason that there are so few clinical trials is that with many of these diseases, we don't know what we need to measure or track in patients to determine the effectiveness of that treatment. And the reason for that is because there is no natural history that has been documented for many of these rare diseases. When you don't know how a disease is going to affect a patient over a period of time, it makes it really hard to figure out what are the best ways to treat it. So now that we have established what natural history is, what it does, and why it's so important, let's spend some time discussing how we collect information about the natural history of a disease. I'm going to discuss three primary approaches that are used to collect natural history data. The first is conducting a natural history study, specifically designed to collect natural history data. And in most natural history studies, it involves the collection of clinical data from patients that were derived from either case histories or clinician visits. Another way to collect natural history data is through a patient registry, which usually serves as a database of information collected from a patient with a specific disease. Finally, you can also collect natural history data through the use of a biorepository, where patients submit samples of blood, urine, or other tissues to be evaluated for genetic or metabolic markers. So, as a way to start distinguishing between these three data collection methods, I will say that, as I alluded to before, natural history studies are designed to specifically collect natural history data. In contrast, whereas patient registries and biorepositories can be great resources to collect natural history data, they can also be used for other purposes. For this module, we're going to be focusing primarily on using natural history studies and patient registries as a means for collecting natural history data. Before we get into the details of natural history studies and patient registries, I want to take a moment to make sure that there is clarity between what a natural history study is and what a clinical trial is. So, in simple terms, a natural history study is not a clinical trial, and a patient registry is certainly not a clinical trial. A natural history study can be regarded as a type of clinical research, but it's not a clinical trial. And the reason for that is because natural history studies are observational, which means that a subject participating in a natural history study is not going to be asked to change anything about their everyday life or the current management of their condition. This is in contrast to clinical trials that are, by nature, meant to be experimental. Clinical trials test the impact of new treatments, so in these cases, subjects are usually asked to change some aspect of their life, such as take a pill every day for a period of time. In fact, natural history studies are often part of the first steps necessary before a clinical trial can occur to ensure that what is being measured or studied in the clinical trial is valid and relevant to the patient population that's participating in that clinical trial. Natural history studies can verify that study outcomes for a clinical trial are feasible and reasonable for a specific disease and for that patient population. And while we are talking about current treatment regimen, I also want to emphasize that although the official definition of natural history includes the course of a disease in the absence of intervention, a person participating in a natural history study is not expected to stop their current treatment regimen if they enroll as a participant in the study. In fact, in the context of clinical trials, it's actually better for a subject enrolled in a natural history study to do exactly what they do in their current treatment regimen, because when that clinical trial takes place following that natural history study, the effectiveness of that new clinical treatment can be measured against what are the current standards of care at that moment, which has more real-life implications. So now that we have distinguished natural history studies from clinical trials, let's talk about the different types of natural history studies that there are. So there are generally two categories of natural history studies: retrospective studies and prospective studies. Retrospective studies look back at already existing data to draw conclusions about natural history. This can be found in, say, literature reviews that are looking at patient cases, but most of the time, existing medical charts on a patient can provide a lot more comprehensive data than a literature review. So that's usually what's used, referred to as a medical chart review. When you are doing a retrospective natural history study, the data that is available, whether it be through lit reviews or through medical charts, that's included in the study, all of that information wasn't necessarily collected with the intention of contributing to natural history, so there may be some limitations in the data itself. But I will say it is easier and usually cheaper to do a retrospective study, a chart review, compared to a prospective study. So a lot of times, patient populations will opt to do the retrospective study first, gather data, and then use that information to inform the design of a prospective study. So when we're talking about prospective studies, these studies were designed with the specific intention and purpose of collecting natural history data. Data can be collected at one point in time, which is referred to as a cross-sectional study, or at multiple points over a period of time, which is referred to as a longitudinal study. And this is usually, once you decide to do a prospective study, this is usually the type of study that's done, this longitudinal study. The data collection usually takes place as a visit to a clinical site where the research team that's running the natural history study will gather data on a patient during that clinic visit. So that's a little bit about natural history studies. Let's move on to discussing patient registries. First, let's distinguish between a natural history study and a patient registry. And in my mind, there are a few differences between the two, and it has a lot to do with the scope of each. First, natural history studies are designed to address specific research questions about the natural history of a disease, whereas some patient registries may have objectives independent of natural history data collection. So some common applications of patient registries that are not explicitly intended for natural history data collection might include being used as a database to inform a patient population of a new clinical trial that they could potentially enroll in, or it can be used to monitor the outcomes of a new treatment that has recently come to market. And to that point of continuing to distinguish between the two, natural history studies are generally led by clinicians and researchers, whereas patient registries can be run by a clinician or a researcher, but they can also be maintained by a patient advocacy organization or even a pharmaceutical company, depending on the purpose of the registry. Another thing is that data that is submitted to a patient registry can be somebody other than a clinician, which is not generally the case with natural history studies. So that data can be submitted by the patient themselves or an observer that sees the patient every day, like a family member. Finally, natural history studies are usually focused on clinical data collection. Patient registries can collect clinical data, but they can also collect a lot broader information, like, say, the daily life impact of a disease on a patient, overall quality of life, or other related assessments. And sometimes that data collection simply takes place on the computer with the patient entering information about themselves as if they were taking an online survey. So let's distinguish between these types of registries. As I mentioned before, patient registries, as one example, may be used just as a potential contact database so that enrolled participants in the registry can be informed of ongoing or new clinical trials that they could potentially participate in. These contact registries may contain some demographic or personal information about the patient, but beyond that, these registries don't collect much more than that contact information so that they can be updated on potential new studies that they could participate in. A registry that collects information on a specific disease, which I'm referring to here as a disease registry, but I'm not really sure if that is the official name. I just want to distinguish it from the contact registry. These disease registries, they can collect natural history data and even can collect clinical data. They can also collect data on patient-reported outcomes, quality of life assessments, as well as performance-related assessments. This information that's contained within a disease registry can be reported by a clinician or by the patient themselves, or, as I alluded to before, an observer that's close to the patient, like a family caregiver or a family member. This data, as I mentioned, can be used for the purpose of natural history, but sometimes there may be uses for it outside of natural history. As an example, sometimes pharmaceutical companies will maintain a patient registry that collects information on disease-related outcomes after a drug has received FDA approval to observe long-term outcomes in patients. So that's just meant to serve as one example of a type of patient registry that is not necessarily directly used for collecting natural history data. As you probably already know, I like to include real-life examples of some of the concepts that I'm talking about in these modules to demonstrate the application of these concepts. So, although I said earlier that most rare diseases have little to no documented natural history, Duchenne muscular dystrophy is one of the exceptions to that point. Duchenne muscular dystrophy is an X-linked disorder that's caused by a pathological variant in the gene for the dystrophin protein, which results in skeletal and muscular abnormalities that significantly affect overall motor function. So since the early 2000s, a research team called the Cooperative International Neuromuscular Research Group has been collecting data on patients with Duchenne muscular dystrophy, so that by 2016, this group had comprehensive longitudinal information on a number of assessments on over 400 Duchenne muscular dystrophy patients. And in the rare disease space, 400, that's a lot. I won't go through all of the assessments, but I've listed many of them here. I will share that a lot of these measurements were originally derived from a retrospective medical chart review that later translated into data that they collected from their prospective study, like vital signs, medication usage, cardiac function, x-rays of the spine. They also had patient-reported outcomes collected as part of their patient registry. For example, they had patients that took the PEDS quality of life validated survey. They also stored samples of blood for genotyping and identification of new biomarkers as part of their biorepository efforts. So this should give you an idea of how rich this data collection was. They used all of these data collection strategies to collect information on patients with Duchenne muscular dystrophy. And now that I've shared these details on the natural history data collection for this disease, let me share with you the outcomes of this effort. So what I have embedded here is a news piece discussing the FDA approval of one of the first gene therapies for Duchenne muscular dystrophy. This piece shares the story of two patients that received this newly approved therapy. I believe this news piece is from 2023. So it shares these patients' story of this newly approved therapy and what impact it's had on their lives and their families' lives. So please feel free to take a look at it at your own time to get a full picture of the impact of what thorough characterization of natural history can do. So as I close, I want to reemphasize a couple of things. Once again, most rare diseases do not have a well-characterized and well-documented natural history. We know, for example, we know a lot about adult refsum anecdotally, but there is still a lot of work to be done to be able to say that we have a comprehensive understanding of how adult refsum manifests throughout the course of an individual's life. And not having that clear picture makes it really difficult to develop targeted new therapies. And this is the case not just for adult refsum, but for many rare diseases, and probably contributes considerably to this understanding of why 95% of all rare diseases don't have an FDA-approved treatment currently. And I haven't really focused too much on the role of patient collaborators in this module as I have in previous modules, but it's important to state that patient collaboration is a crucial part of gaining insight on natural history. Who can speak better to how a disease manifests over time than the person who lives with this disease every day? So you, as a patient collaborator, You hold so much knowledge, experience, and insight that can be tremendous contributions to the understanding of natural history of adult refsum disease. And the better the adult refsum community and all of its stakeholders engages with patients as collaborators, the more information gained, the more information is gained, and all of this contributes in the long term to more targeted treatment options that lead to better outcomes for all patients with adult refsum disease. With that, I will close up. Coming up next, we will talk about the drug regulatory and approval process, and this will shed some light on the steps towards FDA approval for new and repurposed drugs. And this will also include discussion on the different types of clinical trials. So I want to thank you for your time, and I will ask you to take the survey that is associated with this module. I look forward to continue being your guide through these modules. See you next time. If you have any questions about this module or this training series, please contact the Global DARE Foundation.